my view of what the future might look like for medicine. It's science fiction, it's not science fantasy. If it's science fantasy, it would not be real. If it's science fiction, it's an extrapolation of what we have today to what might be developed in the future. So it's where medicine might be going, where we might see some things happening, and how much, but it's done from a proteomics, multiomics, informatics, AI perspective. So let's see what we think. You're the people who are going to have to develop this, by the way. No pressure. So it's a statement by Sir William Osler, who was quite advanced in his time. This is 1892. And he said, if it were not for the great variability amongst individuals, medicine might be considered a science and not an art. Any medics in the room? I have had a few people throw shoes at me for this statement and get quite upset. But what it's meaning is medicine, as it was practiced then, and we've gone a long way since then, is based upon observation of symptoms. It's not based upon the molecular characterization of a disease. They just didn't have the technology. With the post-genomic era and with the advances in various multiomic techniques, we now have the potential to define a molecular symptomology of disease. So we can look at molecular profiles and understand those molecular profiles in order to determine the causality but also the heterogeneity of disease. So, medicine is and was based upon observation. And the descriptors, so the nomenclature, for want of a better word, of medicine, is based upon the symptoms, the location, the body part. And by the location, I mean the location in the world, not the location in the body. The body part, the person who discovered them, the age in which they occur, the causative organism, which is probably about as advanced as they got then. So medicine, there's no standard way of defining these things then. So do you know about ICD codes? ICD-10. So what ICD-10 codes are an attempt at a standardized nomenclature. It's a code that represents a disease type and disease subtype. So it's a standardized notation for a disease. So at least that is starting to come together, at least from the disease perspective. It doesn't use this loose language that they have. So it's a standardized code. So that will be important because you can use an ICD-10 code, and I think they have also an ICD-14, which digs deeper, but this code defines a particular disease type and subtype. So, but how were they characterized? Well, hypertension's first example, that's a symptom, but people call that as if it's a disease, and actually it's a condition. Japanese encephalitis. Does it mean it only occurs in Japan? No. It was probably prevalent at some point in Japan, but why does that make it any clearer, whether it's in Japan, UK, America, Brazil, whatever? It's an irrelevance. Breast cancer, well, that occurs in the breast. Alzheimer's disease. Why do we name Alzheimer's Alzheimer's? Does it occur in the Alzheimer? No. Big grammatical clue. It's got a capital A and it's got an apostrophe. It was developed or discovered by Alois Alzheimer in Germany. So Alzheimer's disease was discovered by Alzheimer. Senile dementia, well, it's sort of an age group. And amoebic dysentery, well, it's caused by amoebas. But you can see how there's no standardization here. It's all over the place. So the taxonomy is diverse, doesn't define the disease, and it kind of is a bit of a nonsense, really. So this makes it hard to treat. We have a numerical nomenclature in the ICD-10 codes, but really we should also be having a molecular nomenclature for a disease state. Some point in the future we will do, I'm sure. Now, we can start to get to that in the post-genomic era, because now we can start to look at the potential for the characterization of diseases at the molecular level. We can measure the disease in a molecular basis. There's problems because there's a lot of information. That's the whole basis of this program to some extent. Diseases are diverse and we have to search through lots of information. Fine, we've discovered that and discussed that. And we should ideally have a small set of features that define a disease from others. So what sort of personalized medicine is there? And this is how medicine may go, and it is going this way. So first of all, we have diagnostics. The diagnostic defines somebody with the disease from somebody who doesn't have the disease. So for example, prostate cancer versus BPH, benign prostatic hyperplasia. We also have prognostics. So a prognostic tells us how somebody's going to do. In a basic form, we might say, what is the probability that a breast cancer patient will undergo metastasis before or after 5 years? More specifically, we can look at how long will it be before. So one example might be, how long will it be before this disease progresses? We predict with our algorithm that it will progress at 11 months. That's a more accurate prognostic. Just one point around accurate prognostics, you have to be careful because, yes, you want to identify the risks for somebody, but it's like saying to somebody, you are going to die on the 12th of December 2019, or 2029. How does somebody deal with that information and process that information? So you have to be careful about how you present it. Usually it's low risk, medium risk, high risk. You don't tell somebody the day they're going to die, it doesn't go down well. So there's an ethical issue there. And the third type is predictive medicine. And this is where personalized medicine starts to come in. So which individual will respond to a therapy? What's the probability that this individual who has CML will respond to imatinib? It becomes a tool for prescribing, a tool for treatment. So that's the key next step, if you like. Imagine a situation where we fully understand, with companion diagnostics, this is what they're called, the best treatment to give to a patient to maximize their response and minimize their side effects. Now, with the data we have, with the methods we have, and we need to have the buy-in from pharma to some extent, but with those things coming together, we have the potential to start to look for personalized medicine and to give patients the best possible treatment. Now, some cases that exists. So, for example, if in breast cancer a patient is ER positive, we know that we can give them tamoxifen. If a breast cancer patient is HER2 positive, we can give them Herceptin. And then if they're triple negative, we try basically various cycles of chemotherapy. But to give a case in point, those simple predictive response tests have increased the likelihood of survival to 10 years for breast cancer patients by about 80% over the last 10 years. So if you know how to treat the different heterogeneities of a disease, you can massively increase survival and de-risk that for populations. So that treatment and personalised treatment is already yielding benefits, but it can go further. So where are we going with data generation? Well, in all likelihood, we will see an increase in coverage of the proteome and metabolome. So we're getting more data, more depth of data, more accurate data. We'll get an increased depth of sequence of the transcriptome, although that's not got that much further to go. We will get more accuracy with reduced sample volumes. So we'll be talking picolitres or femtolitres of blood, getting a diagnosis. Non-invasive tests. So there are already technologies that, based on putting a patch on your arm, you can draw interstitial fluid in a non-needle based approach. So you could have patches which are taking samples all the time. Yep. The other thing is there is always a trend of miniaturization in technology. You know, when I first had a mobile phone, it was about that big. Well, it's quite big now, but it packs more data in now, but you know, phones went quite small for a while. First computer I ever saw took up a room the size of this room with a little terminal that sat at the front of it. Technology is miniaturizing. So if technology is miniaturizing, can we have a handheld mass spectrometer? Can we have handheld real-time sequencing? If you've seen an ion torrent, by the way, ion torrent sequencing, little cassette about so big. Things are getting smaller. Technology is becoming more compact. Could we develop a real-time ion torrent sequencer or real-time sequencer or a real-time mass spectrometer that gathered data on my blood proteome continuously? Cost will come down, which means we end up with more data, we end up with more frequency of measurements. At the end point of this, can we have point of care diagnostics, so a device that does mass spectrometry, let's say, wearable proteomic devices, or wearable transcriptomic measurement? Now, if you're measuring the whole proteome and the whole transcriptome, then the data you're generating doesn't only answer one question. Imagine a situation where you've got a wearable device measuring your blood proteome, detecting all sorts of issues and risks. You have the potential to monitor risk of 50 different diseases in real time on a daily basis. Okay, your markers are up for this, the risk of this disease. Go and see your clinician. They will find out the best treatment that you have, catch that early, or even give you lifestyle changes to prevent that thing even occurring. So we have the potential to get to this point. In the interim, it's early detection of disease. There are cancer programs now which are looking at people who are being detected pre-symptomatically. Or, if we're gathering data about our molecular profile and our lifestyle, given the pool of thousands of other or millions of other individuals, can we make suggestions about lifestyle that will prevent us getting the disease in the first place? This is starting to sound a bit like Ayurveda, by the way. So prevention is even more powerful than cure. In trends in diagnostics, well, we have early stage diagnostics, but our knowledge is hypothesis based. Often there's a high attrition rate for developed tests. These tests fail quite often. With increased data, we get greater population coverage, we get better temporal and spatial coverage. There's a huge opportunity for diagnostic discovery. This will rise, there'll be a rise in increase of multifaceted diagnostics. So an algorithm that looks at your whole proteome, as I said, diagnoses the risk of 50 particular conditions. Currently, it's one marker for one state. So P53, HER2, ER, single marker measurements, giving one state. Sometimes it's maybe a small number, 1 or 2 or 3. But if we move to a whole proteome approach, we can have multiple markers, multiple states. Now, if you look at most cars now, people plug in a diagnostic device. It looks at all possible problematic states of that car and says where there's a problem. Now, with a particular sample from a human, can we look at all possible disease states and problematic states that might be going on for that human at that point in time, based upon a complete proteomic or transcriptomic profile. Blood is kind of the universal diagnostic fluid because it encounters the whole body. So if you've got blood markers, it's relatively non-invasive and can detect things and check for things. So there's a move to multi-markers, multiple states, a move to prognostic models, early detection of disease and personalised medicine, and we can potentially couple therapeutic development with response to therapy. So this is starting to happen. Drug companies, by the FDA, are required to put in companion diagnostics. That is kind of loosely adhered to at the minute, but if there's the right data, you could actually couple a clinical trial with response predictors in order to be able to develop a personalised or companion diagnostic for that treatment. Trends in AI and data analysis techniques. Where's that going? Well, we are continually getting higher performance of computing. We're getting greater explainability into that. We've got kind of a synthesis of LLMs coupled with evidential approaches. So people are developing LLMs, validating them with real-world data. We've got better algorithms for integration of multiple data sets, multi-omic data sets. We've got development of digital twin models. We've got development of in silico drug response models, virtual patients, and virtual clinical trials. So based upon data, we can simulate the potential of a drug to change a system. I mentioned to you before, DepMap, the data there. That will go further. Trends in treatment and drug discovery. Well, we're going to have more integrated pipelines of discovery. We can model currently biological potential. We will go further and look at the ADME DMPK. So this is about the metabolization of drugs, about the efficacy of drugs, about where the drug goes in the body, about the degradation of drugs, the half-life, whether it binds to fats. All of that can be modeled ultimately. For early diagnosis, we can identify determinants of disease. We can look at preventative treatment. And there's a big move towards aging lately. So we can look at age-related diseases and start to find cures for age-related diseases. And if we know systems are dysregulating, there are mutational forms of genes, can we correct those with CRISPR-Cas? So can we treat the issues arising from accumulated mutational states over life with CRISPR-Cas, give you back the genome of a 2-year-old? Because aging results in it occurs because of rising mutations. It occurs because of telomere length dropping and a whole load of other genetic changes that are occurring in response to your environment. So we can see and expect an increase in gene therapies and also an increase in personalized medicine. An emerging area is synthetic biology. So people are already building virtual cells based upon molecular organization and engineering. So with these, we can build better models of disease. One of the big challenges for many diseases, particularly in neurological diseases, is getting good models of diseased neurological tissues. We're starting to see some of that. People are starting to create much more robust models for neurology. But if we can get good representative models at the cellular level or at the virtual, at a cultured organ level, we can create an artificial cell-based heart which we can then use for treatment. It moves away from animal models which are not representative, but moves towards something which is more representative of a human state. One of the things that's also potentially emerging is the use of stem cells to grow organs on a chip or regrow cultured organs in a vat. So we could grow somebody a replacement heart, liver, kidney, so on, based upon stem cells, hematopoietic stem cells which have been induced with induced pluripotency from a sample. So you can take blood cells, induce iPSCs, differentiate those iPSCs to tissues, use those to create an organ replacement. We will also develop in silico, molecular level, tissue level and organ level systems models and models of the interplay. So you can imagine through having digital twin of a system, we can join all this together to create a molecular virtual human. Which means you can start to do things like create this temporal model. What happens if I change this gene? Suddenly this gene mutation arises in this tissue. How will that affect cascade through the whole organism? There's potential to do that. The guys who who discovered AlphaFold are currently building a virtual cell model based upon all the pathways in a cell and using that to create a dynamic model of a cell. There are limitations. Which cell are they choosing? How do they validate that as well? But there is, again, move towards a virtual digital twin of a cell. And this statement came from the guy who won the Nobel Prize for AlphaFold. He was presenting that at that meeting. So we could end up with whole individual digital twins. I give the system your genome, and it will consider a version of you which is a digital twin of you, which is generated through AI, but also through systems level models. So we can then look at experimental treatments on the digital twin of you. How will you respond to this drug? Can we, run a scenario where we give you a treatment without you actually even ever having the treatment to see whether it cures a problem? Does it cause problems down the line? Are you going to have an adverse reaction to this drug if we give it to you, based upon you as a digital twin? So that's quite radical, but it's kind of a view from my perspective. I'd be happy to answer any questions, but feel free. If you've got a radical disagreement with me for any of this, please, I'm quite happy to hear what you have to say. Have I stunned you into silence? Is that what it is? Any questions?
Just a hypothetical question.
Yeah, well, this is whole hypothetical, and that's fine.
At our level, like not with so much advanced techniques and so at what level we can go to design a, not cellular level, but at least a few combinations of pathway interactions, like a whole model, like how we can do it?
Yeah, so the method I showed you in Weka, if you wanted to look at how things are interacting and how stable they are, the method I showed you in Weka, where you're mining across a whole load of things and getting weights out of those things, so you're getting enrichment and then you're getting weights out of the Weka model, you could use that to start building an interaction model of a system.
Suppose we are giving a transcriptome data, like we will be giving it in a raw form or after annotation?
So it doesn't matter how you're giving it. Say you've got a FASTQ. Well, that's the raw form. If you know which version of the genome library it was run under, then you can run that through Galaxy to get gene level information, transcript level information as TPM values. So if you go to the raw form, you can use that. Usually it takes about a day on Galaxy to run that. So you've then got the normalized gene length corrected data for that human. Once you have that, then let's say you've got a particular hypothesis or question you're exploring, then you can mine that to enrich the features associated with that question. And then maybe there's a different state, maybe there's a mutational state, then you can mine that to see the features associated with that question. And you're getting gene lists each time. Then what you want to do maybe is look at how those genes predict one another.
Maybe like checking the small molecules sensitivity.
Yes, yeah, so you could, in that enriched list, maybe it's 500 genes, maybe it's 100 genes, running that through Gnome AI, drug Gnome AI, running that through Kemble, running that through Reactome. All of these, from this list, then you've got all sorts of other options to explore where those genes are, what they're doing, how you can interact with them, how you can drug them. If you want to create siRNA against them or CRISPR-Cas against them, you can find the mechanisms of that very easily in gene cards. So you've got it's like a tree. The first branch of that tree is which genes are associated with this question. And then you've got all sorts of different routes you can go down to explore what they're doing, what the context of them is, which pathways are they in, which drugs target them, and so on. And that's really what all this is about. It's about workflows going through from data. So there's nothing really to stop you doing anything. If you've got data, or you've not got data, you go get public data, you can start to explore all of this. You don't need a lab, all you need is a computer. Yes, you need a lab to generate mass spec data, and you'd need a lab to do RNA-seq. But in the absence of those, there's a lot of public data out there which you can try things out on. So if one thing I would say to you is go out, look for data, have a play with that data, try some of these things out, and start exploring, start experimenting. At the end of the day, you're running experiments, you're just not running them in a test tube, you're running them on a computer. And with this course, you should have the components necessary to do that. But if you get stuck, email me. If there's a question you don't understand, how do I make this step? I've got these things, where do I get to next? Let me know. Any other questions? So you're going to now change your proteome by having lunch.